Today I'm going to be talking about dynamic cognitive assessment in health and disease. Since this is only a 10-minute presentation, this is really mostly a teaser. So, touching on a little bit of methods and how we develop digital cognitive assessments, and then really getting in a little more into what it enables in terms of understanding change over time in cognitive functioning. Disclosures, I have two roles on two 501c3s, and then the funders who have enabled this work. So digital neuropsychology is a shift, right? It's a shift in the way we think of neuropsychological function and the way we think about cognition, not just because we're translating methods from primarily paper and pencil to digital tools, but also because of the new types of measurements and new ways of thinking that this potentially enables. So, three general considerations that I like to think about when I think about the development of digital cognitive assessments is, first, psychometrics, so building measures that are sensitive, reliable, and efficient. So measures that can be done briefly and still yield good data and meaningful data for looking at differences between individuals as well as differences within an individual over time. Engagement, so I'm not going to get on this too much, but in a digital environment, engagement is a huge challenge. Thinking about things like user-centered design and universal design principles, but also how things like return of research results can help bring people into digital cognitive assessment in a way that is hard to do otherwise. And so through testmybrain.org, we've done a lot of work in this approach and have tested about 2.5 million people since 2008. And then finally, technology. So thinking about differences between devices, the demographics of device ownership, how this affects cognitive test scores, and then how changes in devices over time really mean that our science has to change and continue to evolve as devices evolve. They use these devices. So looking at it from these three perspectives, the way we develop measures is, fundamentally different than the way you might do it when you are looking at a paper and pencil approach. So this is software. So we take a software development approach to developing measures. So the idea is starting with test prototypes, doing lots of A/B testing, which is just randomization essentially, to evaluate not just psychometrics, but also engagement and impacts from the technology themselves in order to build tests that are really fit for digital assessment, fit for widespread use in groups that are demographically diverse and diverse in the environments that they're doing testing in. Again, this is more of a teaser than anything else. I refer you to a paper that we recently published in Neuropsychopharmacology that goes into these sorts of approaches and the implications for assessment and the development of science. But dynamic cognitive phenotypes is what I want to focus on today. So the idea that, although we aim to capture best performance, and that is sort of clinically the way we think about neuropsychology and cognitive function, but this ignores meaningful variability in information processing. And so, the idea that cognition is static, or that insofar as it varies, those variations aren't important for understanding clinical phenomena or, everyday adaptive behavior, is one that's largely unsupported. So this is another case where having a limitation in your method is reflected in the science in a way that really we are no longer in a place to have that limitation. So with digital devices, we can now assess in a way that allows us to incorporate a temporal perspective. So what I'm showing you here in this figure, this is mock data. The idea is that you can have three people who look largely similar from a mean performance perspective, but have differences in variability and differences in the sources of that variability that are potentially clinically meaningful. So I'm going to present some data to you today from the GluCog study. This is a study of people with type 1 diabetes, and type 1 diabetes is characterized by a loss of insulin-producing cells, which results in high blood glucose. And then the treatment is insulin therapy, which results in low blood glucose. And since the brain is highly glucose sensitive, these fluctuations in glucose potentially cause cognitive variability. And we know that causes cognitive impairments when people are experiencing hypoglycemia, especially prolonged and severe hypoglycemia. So the design of the study is that we have 200 adults with type 1 diabetes who are, the study is ongoing. They have a clinic visit. We do baseline assessment at home, and these are these sort of full-scale cognitive measures that are, more similar to what you see traditionally in research on cognition and neuropsychology, as well as measures of sleep, mood, and anxiety. And this is followed by about 2 weeks of ecological momentary assessment. So here we're doing brief measures of cognitive function, asking people about their mood, about sleep, about stressors, and we're getting glucose readings every 5 minutes continuously through this 2-week period. And this is through this device that you see here, which is the Dexcom G6. It's a continuous glucose monitor that measures interstitial glucose, which is highly correlated with blood glucose, but with a little bit of a lag, about 10-15 minutes. And this is a blinded study, meaning that participants do not see their glucose as it is being read by the sensor. We have 3 cognitive measures that we're using here. So one is a digit symbol matching test, which is a measure of processing speed. One is the gradual onset continuous performance test, or the GRAD CPT, which is a measure of sustained attention and response inhibition. That is to say that it is only 45 seconds, but it is a very dull 45 seconds. And so you have to really concentrate to do well. And it's very fatiguing. And then multiple object tracking, which is a test of visual, spatial, working memory. And so you have to maintain these dots in mind as they move around the screen and are occluded. We have baseline full-length versions of these measures, and then we do them 30 times over the next 10 days. And the current analysis I'm going to show you are based on 20 of 200 participants. And as I said, the study is ongoing. So what do you get when you move from a static to a dynamic viewpoint of cognitive information processing? So I think the first thing and the most obvious thing potentially is you avoid temporal sampling error, right? So the idea that, at this time point, this represents your cognition, when actually at another time point, you're doing very different. You have very different performance. So what you see here on the y-axis is response impulsivity. This is a criterion measure. So in this case, higher values indicate more impulsivity. And for participant 1, their baseline assessment is really consistent with the mean across the EMAs. So here's one where, temporal sampling error at the baseline was not a huge factor. And for participant 2, however, we see something very different. So this person has a much lower score at baseline, so much less impulsivity response-wise at baseline than they did in the following EMAs. And here, if you took this one time point as representative of this person, you would potentially underestimate to the degree to which they are impulsive in an everyday environment or when tested repeatedly in an EMA type framework. Temporal sampling error has implications. So if we look now at the correlation in people's baseline scores with age versus the average across their EMAs with age, we see that the averages in case of all these tests are more highly correlated with age. So the correlations are larger in magnitude, negative because age is associated with poorer cognitive ability. So this gives you a sense that, that average might be more valid than the single time point baseline. Longitudinal change. So we often talk about practice effects in EMAs, and those practice effects themselves potentially could be signal that's meaningful and interpretable. So participant 1 has basically no practice effect across the course of this 2 weeks. Participant 3, however, is showing a decrease in response times consistent with faster processing speed as they go. Mean versus variability. So these are 2 participants with roughly the same mean performance, or same response impulsivity in this case. However, participant 4 has roughly twice the amount of variability in their scores than participant 1 has. So it could be because of things that were happening in participant 4's life at the time, or it could be that their cognition is simply more variable and less consistent. Variability often precedes decline. And so, variability might be a more sensitive marker of differences in neurological functioning and ultimately long-term cognitive change. And then finally, physiological stressors. Sorry about that stray word time on the right. This is a participant who we noted on the GRAD CPT. This is the discrimination score on the GRAD CPT, so how well they were able to perform, essentially. They had one score around time point 14 that was pretty low for them. So these shaded areas are representing plus minus 2 standard deviations from the person's own mean. And if you dig in, let's look at their blood glucose now. This is their blood glucose. This is their glucose measured by a CGM over the course of the study. The blue area is healthy levels. So a normal person would really only vary in this range, but this is a person with type 1 diabetes. So we see big excursions into high glucose or hyperglycemia, as well as some low blood glucose here, or low glucose, which is values under 70. And then one time point where they had pretty severe hypoglycemia. And in fact, if we superimpose these, the time point where they had severe hypoglycemia matches up with the time point where they did pretty poorly or had lower performance on the GRAD CPT based on discrimination scores. We also see this sort of hypoglycemia hangover, which is often anecdotal, but we don't really have the kind of studies that can have someone sit around for 6 to 8 hours to see how they do after they've had a hypoglycemic episode, much later. But we do see lower performance in the period following hypoglycemia as well. So this matches exactly what you would expect if these measures were sensitively getting at differences that were related to blood glucose. So, conclusions, none. Stay tuned. This is an ongoing study. And I think a lot of this work is in the early stages, but, hopefully we're getting to a point over the next 5 years where we can really start measuring a lot of things with digital devices. So cognition, but also things like, motor functioning, mood, affect, thought, speech. Probably a lot of other talks in this session will be dealing with these sorts of phenotypes and measurements. But from the cognition perspective, I really hope we get to a point where we have a neuropsychology and a science of cognition that is dynamic rather than static, and also that's context rich rather than context free, because we know that people exist in environments, they exist in ways that are, our lives are inherently dynamic, and the fact that our cognition is also dynamic should be the default assumption. So that's all I've got for today. Acknowledgements, the GluCog team, my co-PI, Naomi Chater, as well as folks at other institutions, the folks in my lab who have helped with a lot of the development of tests and analysis, Many Brains Project, people at McLean, particularly Institute for Technology and Psychiatry, and the funders that support this work, so in particular here, NIDDK and NIA. Thank you.